It's been such a pleasure to be part of this collaboration over the past years. As you'll see from this talk, it's been very influential on me, and I'm very grateful. So many of you know that I am a memory and vision researcher. That's how you've known me. That's what I've studied up to my career at this point. I'm in the process of redirecting my entire research program to study a very different brain function, and that is mood. And this is one of the first talks I've given in this space. So really excited to tell you today about the representation of mood in the primate insula. For those of you who haven't had neuroanatomy in a while, let me remind you where the insula is. So here I'm just showing you a little plastic model of a human brain where I've removed one of the hemispheres so you can see the insula tucked deep down in there. Insula is cortex, but it's tucked deep in the brain and it has its own little lobe. And a number of lines of evidence implicate the insula in the awareness of feelings, including mood. So stimulation of the insula can evoke ecstatic experiences, and damage to the insula can cause emotional blunting, including problems with mood. So clearly, there's some fascinating stuff going on in this little brain area, and the big question is, what's happening in there? So today, I hope to shed some light on the answer to that question. Before I dive into the science, I just want to tell you a little bit about what's motivating this pivot for me. So for the past handful of years, I've been involved in writing this book. It's called Elusive Cures. It will publish next month. And in the book, it was really a personal journey for me to ask and answer the following question. What is it going to take for us as a community of brain and mind researchers of all types to traverse from where we are right now to an understanding of the brain and mind of the type that will be required to inform new treatments for brain and mental disorders of all types, but particularly the most formidable disorders, so our neurodegenerative conditions, our neuropsychiatric conditions, and our neurodevelopmental disorders. And writing this book was an absolute transformative experience for me. I learned so much in this process. I don't have time to tell you about all the things I learned today, so I just want to focus on the one that's motivating my pivot. And that is, if we zoom out and just ask as a community, let's take stock. How are we doing at understanding the brain and the mind? There are certain classes of disorders that are progressing at a rapid clip, undisputably. Things like vision and memory and motor control, all the great talks you've heard at this meeting. But there are other classes of disorders that are highly relevant, particularly to the neuropsychiatric space, where progress isn't happening quite as quickly. And they include the neuroscience of many feelings, including mood. And the reason these aren't progressing as rapidly is because they have special challenges that are really, really hard to overcome. And we need solutions to those challenges desperately. Because, after all, we know that a remarkable 20% of adults will experience a mood disorder at some point during adulthood. And while we do have existing treatments, they don't work for everyone. And so we need a neuroscience of mood to progress, to help understand and develop those treatments. So what are these special challenges? Well, if you just take a step back and you ask, well, how do we understand anything? A lot of the functions that we're used to working with, they have an objective ground truth to them that we use as a benchmark. And around that, we can shape a task. For example, if you want to study memory, like I used to do, you can design a memory task, and there is an objective right answer to the questions you ask, like, have you seen this before? The challenge of mood, when you ask questions like, how happy are you, is that it is a subjective experience that has no objective ground truth. No one knows how happy you are but you. And this leads us to the following problem. In humans, we can probe subjective experience, but we can't get high-resolution, densely sampled measures of neural data, and we can't deploy this suite of new tools we have to probe causality, like optogenetics. In animals, we can make those measurements and do those manipulations, but we can't probe subjective experience, at least not to the same degree. And I'm convinced that in the case of everyday mood, there really aren't great behavioral measures. There won't be great behavioral measures for it. In cases of extreme mood, like the ones Austin Cowley studies when he studies anhedonia, I think you might get something out of facial expressions, but I'm not sure that this is going to work for everyday mood. So we need to get through this bottleneck desperately. And as we do, I think it's important that we kind of keep our eyes on the prize and ask, well, what is it that we're trying to understand to begin with? So a systems neuroscience of mood would describe how the good and bad experiences that we have are transformed into what we perceive as moods. At the heart of mood is the notion of integration. So mood is often distinguished from the other emotions, and that emotions are triggered by things. So you see a tiger, and that triggers fear. Or you see feces, and that triggers disgust. But in the case of mood, they're often described as much more diffuse and slowly changing. And everything I can see suggests that this is because moods are not triggered by individual events, they're an integration across many experiences. That is not to say that the other feelings aren't integrations. You'll hear in the next talk, I imagine it will be fantastic, by David Anderson, where he'll talk about integration and persistence for other more primal emotions. But in the case of moods, it looks like the integration is turned up to 11. Okay. And so a big question in the systems neuroscience of mood is where and what is the integrator? So with all that in mind, we set off to try to find the representation of mood in the primate insula. This work is a collaboration between my group and the group of Veit Stuphorn, who is at Johns Hopkins University. He has a very special data set. It's one of the only data sets collected from monkeys who are performing behavioral tasks from the primate insula. And the original collection of this data set was led by Yuping Yang, who was a postdoc in Veit's lab at the time. She's now a faculty at the National Defense Medical Center in Taiwan. We also have contributions from Katrina Hacker from my group. So we're going to take a two-step process. The first step, we're going to create behavioral models of how happiness fluctuates, and then we're going to use those models to probe the neural correlates of happiness in this data set. So step one was motivated, it's really directly inspired by what I consider to be a near genius move by Rob Rutledge about a decade ago. So Rob was building on the observation that a fantastic mood induction paradigm are the risky decision-making gambling tasks that so many of us know and love. The idea is that when we win, we get happy, and when we lose, we become less so. And Rob saw this as a really excellent testbed to describe models, behavioral models, of how experiences are transformed into moods, because these tasks are quite easy to parameterize. So I'll show you how this works, not for Rob's task, but for the version of the task that we're using. So in this task, there are two types of rewards. The subject is accumulating token rewards to get concrete ones. In the case of humans, those concrete rewards are money. In the case of the monkeys, that's juice. So on each trial, this is the human version of the task. The subject has to choose between a certain reward or a gamble. So here the choice is I get 1 token for certain, or I get to take a gamble. Well, I'll get either 3 tokens or 0 tokens with 50-50 probability. The subject chooses the gamble. The outcome is revealed. Yay, you won. 3 tokens are added to the total. And when the subject accumulates 6 tokens, they get 50 cents. So in different trials of the task, they will either win or lose tokens. And the number of tokens and the probability of the gambles changes from trial to trial. Now here's the simple but genius move. Every 2 to 3 trials, Rob inserted the question, how happy are you at this moment? Rate that on a continuous scale. And here's a plot of happiness as a function of how many times that question was asked as the subject played the gambling task. And you can see happiness is fluctuating up and down. Now the goal here is to build a model of what is driving the answer to this question of happiness. I'll just jump to the punchline and show you here's the model. I think you'll agree it's a pretty reasonable reflection of the subject's happiness variation. And so the question is, what is the model? So the model is inspired by this. There's a correlation between those subjective happiness reports and 2 types of rewards, not just based on what happened on the past trial, but an integration or a correlation across several trials back, with concrete rewards mattering more than tokens. So this inspires a model in which happiness is a weighted sum of the 2 types of rewards, where concrete rewards matter twice as much, as an integration across these trials, about 7 trials back or so. So for a subject, we can take the wins and losses they experience in the gambling task, pass them through, create this weighted sum. You get 2 traces, one for concrete rewards, one for token rewards. You sum them together, and that's the model of happiness. Okay. So this is somewhat happiness demystified. What is happiness? Happiness is integrated reward in this model, integrated across time and also across these two types of rewards. And this is a pretty good model for this subject. Now, simpler models don't work as well. So if you try to model happiness just based on congregate reward or just based on token reward, or you don't integrate, these models don't do as well either for the subject or across the population. And we can come up with more sophisticated and complicated descriptions of what happiness depends on based on things like expected value and reward prediction error. Those models do a little bit better, but they're also much more complicated. And I think it's important to be very conceptually straightforward in this space. Happiness is integrated reward. We can talk about more complicated versions. Okay. So we now have a model that will predict how anyone, I should say, the other thing we're doing different than Rob is we are looking for universal models of how happiness will fluctuate. So the fluctuations that are common among all of us. And again, it's not rocket science. We all get happy when we win. We all become less happy when we're sad, and we all integrate. So we fit the model to 43 people, and we use that to predict the 44th. And that's what you're seeing here. It's remarkable that it works. And it works so well. Okay, so now we have a model that will predict how anyone's happiness will fluctuate as they play this task. And we can use that to study the neural correlates of happiness in the brain. So for this next step, I just want to remind you that happiness is different than the other brain functions you're used to thinking about, right? It has this special bottleneck. It has this special set of considerations. We're going to have to solve this in some special way. All the solutions are imperfect. We're going to do something that it's non-traditional. Some might call it a little bit scandalous. I like to think about it as high risk, high reward. We're going to do a cross-species comparison here. So we are going to use this model to estimate how happiness is fluctuating in the monkey brain. Specifically, monkeys are performing the same task with a few modifications. So instead of numbers, they have symbols they've learned. Instead of money, they get juice. But we can take their wins and losses, pass it through the model, and estimate how happiness would fluctuate as they play this task. I want to emphasize that when we do this, we are not hypothesizing anything about the monkey's subjective experience. As far as I'm concerned, that's unknowable. We have no access to that. What we're hypothesizing is that this quantity is computed and reflected in the monkey brain. And this could fail for all sorts of reasons. It could fail because this is a cross-species comparison. It could fail because what's motivating this work is the question, how happy are you? And we're not asking the monkeys that. Maybe you don't even see this in the brain area we're looking in, the insula, if you don't ask. But if we see strong brain behavior relationships, we might be onto something, and onto something impactful. What I'm about to show you are brain behavior alignments that are as strong as anything I've seen in my entire career of dealing with data sets. And as you know, I've been dealing with the visually evoked response in visual cortex for a few decades, right? So let me show you. Okay. So we were motivated to look in the insula based on fMRI. That's where it pointed to in Rob's experiments. So here we're looking at the period between the trials of the monkey's gambling task. With the rationale, that's when we would have asked the question, how happy are you? And here, so this is 0 is the onset of the next trial. And so we're looking at the 1 second period before. Here I've grouped trials into quartiles based on estimated happiness. And you can see this neuron has a strong and persistent representation across this period, where as happiness is higher, the firing rate is higher. If we look trial by trial, there's a plot of estimated happiness versus spike count. Each dot here is a different intertrial interval period. And you see a crazy strong correlation between these two quantities. It has a Pearson correlation of 0.53. If you look now at this neuron's correlation, there's no model here. This is just the correlation between the neuron and the events that happened on previous trials. This neuron's firing rate is correlated not just with what happened on the last trial, but both types of rewards across a handful of trials, like the behavior. We find many neurons in the insula like this. Bill, reviewer number 2, would love this study. I went into this with a population-based approach, and I realized, wow, this is reflected in individual neurons. It's crazy. We also find neurons like this. So these are neurons whose responses are lower when happiness is higher. They're negatively correlated with estimated happiness, but they also reflect not only, their firing rates reflect not only what's happening in the last trial, but several trials back, or a handful of trials back. Altogether, in a population of 224 neurons, we find about 40% of them are significantly correlated with estimated happiness. We find fourfold more positively correlated as compared to negatively correlated neurons. If we line up those traces and flip the sign for the negative ones, so these are just the significantly modulated units. So we have a correlation magnitude versus what happened on previous trials. You can see we get these two traces. So this is the neurons in the insula. This is the correlation with behavioral reports of happiness. You could see these two bear quite a resemblance. And finally, if we line all of the neurons up into a pseudopopulation to estimate the overall vigor of the insula population response, whether a neuron was correlated or not, we get a correlation between the grand mean firing rate of the insula and estimated happiness of a Pearson of 0.71. So this is what I mean by, this is one of the strongest brain behavior correlations I've ever seen. A few years ago, we did some work in the visually evoked response in visual cortex, looking at another parameter. So this was IT that modulates overall population response vigor. It doesn't matter what it is. It's called memorability. But we found a Pearson correlation there of 0.69 with the firing rate. But this is the visually evoked response in visual cortex. This is the behaviorally unconstrained period between trials of a monkey's task. And the question is shaped around how happy are you? Right? I find that this is remarkably strong in that light. And finally, going back to this, we also wanted to know the question, is this something that's reflected ubiquitously across all higher brain areas, or is this something special to a subset of them? So we looked in dorsolateral prefrontal cortex, also known for its persistent activity, and there we don't find a strong representation. So the pseudopopulation doesn't have much going on in it. And as compared to this number 38 here, the number is 9%. Okay, so taken together, the big question is how are experiences transformed into mood? Here, this version of mood, happiness. We know 4 things. We know that happiness is reflected in the insula as persistent activity. We know that it's integrating events over about 7 trials. We know that it's integrating over 2 types of rewards, so a second type of integration. And we know that it's reflected via combinations of positive and negative neurons. So to us, this is very reminiscent of an emerging sketch of how this integration is happening, one that you've seen before many times in the SCGB collaboration, which is a reciprocally connected population of neurons that are both excitatory and inhibitory of the type that Lim and Golden introduced, and you've seen by so many others. So the big question for us is this happening in the insula, or is it happening somewhere else? And I think that's one of the big questions of mood. So we are currently building on, we know the nodes of the mood network in the primate brain, and our colleague Bijan Pasaran his lab has even gone in and electrically stimulated these nodes to figure out who is the sender and who is the receiver. What we currently don't know is what's actually happening in those brain areas. And so we're beginning to piece together a model, where with Veit we've pieced together 2 of these nodes. We'll piece together 2 others with another collaborator, Deo Lee, 5 others with another collaborator, and so on. But our goal here is to build up a big primate brain model of how experiences are converted into happiness that we can then go and test in humans and can be a first test bed for informing the next big step in mood research. So I just want to end by thanking the Simons Foundation. I'm also, in addition to being part of the SCGB, I'm proud to be a Pivot Fellow, where this transition from memory to mood is supported by this fellowship. And for the next year, I get to embed in the lab of Yael Niv at Princeton to learn more about this space. So thank you very much.
Very cool. Experience affects happiness. Another intuition about happiness is that it affects our behavior.
Yeah.
And have you looked at that in the humans and the monkeys to see if the estimated happiness influences the choice on the next trial? And can you use that to try to get to mechanisms?
Yeah. A lot of definitions of mood are of that type. So they're not actual behavior measures of like what kind of facial expression does it induce, but how does it affect the behavior of another type? And so I'm very interested in those definitions. One of the big motivations for me to study mood is its big reciprocal interaction, and I think this is the next generation of systems neuroscience. So yeah, mood impacts our percepts of the things that happen to us, and those in turn impact our moods and our behavior. So there is a big reciprocal loop of which I've only focused on one half. But yeah, many others who are studying that other feedback loop, and I think trying to figure out how to integrate those two together is
But you have data, right? Because you can look at choice for a particular trial
you're talking
type
about in the experiment,
when
yeah.
you're estimating that happiness is
now I
high or
understand.
low.
Is
And that
this
could be a way
yeah.
to validate it in the monkeys too, if it looks the same as in humans.
Yeah, sorry. Now I understand what you're asking, not you're asking within this experiment. We haven't done that yet, and we should. Absolutely. Thank you.
So I have two questions. One is that are there pattern autonomic responses that you can find in humans that you can use as a behavioral readout of measures of happiness to as a sort of a additional because right now you have no way of asking the monkey, but perhaps a pattern body response can be one way to know that. And second question is, you only showed the intertrial interval sort of baseline activity, which also, CAR-T growth are not have found like in the amygdala. Do you actually see at the end of the trial how that update happens?
So to answer your first question, I am not aware of current validated measures, but I completely agree with you that those need to be developed in humans, like to the degree to which we're going to find some sort of measure. People have looked, and I haven't found them yet, but we're in the era of maybe there'll be some hope there to find something, but there aren't yet any good measures of that that we can leverage. To answer your second question, so I think what you're asking is, how does what's happening during the period within the trial, how does that influence, the new updating rule? And that's exactly what we're diving into in the data set right now. And we haven't made it any farther yet than like a bean counting type of thing. So I can tell you if the neuron has a significant correlation with happiness in the intertrial interval period, this was its population response. I can show you a big messy matrix. So now we're looking for some more coherent description of how those experiences are transformed into mood. So yeah, we're definitely working on that for sure.
Fascinating. If you extend the intertrial interval to arbitrary durations and keep recording, can you measure decay dynamics for these individual neurons? And do they predict subsequent responses when you resume the trial?
Yeah, that's an awesome question. That didn't happen in this data set, but it might be in some data set of the type that we're planning to build upon. It's a great question. Yeah.
Cool. So I'm sure like every monkey trainer, you've had the experience where they have these occasional moments of frustration, sometimes boredom, but often early. And we sort of all are trained by our monkeys to engage in this paradoxical thing that seems like the wrong thing to do, which is you just give them a bunch of free reward for doing nothing, right? Which is like standard operant conditioning. This should be the opposite of what you should do. You're rewarding them for giving up. And yet we all know you give a monkey a bunch of free juice, they'll perk back up, they'll overcome their frustration or boredom or whatever it is. Mm-hmm. So do you see that activity in the insula, for example, you must have these moments in your experiments where the monkey has a bunch of failures and just gives up. Like, are those moments when the insular activity is low, when you give them reward, does it perk back up?
Yeah, no, it's a great question. Anecdotally, I did not do these experiments, but I could ask you ping that question. Yeah, I don't know. It's a great question. Would that be recorded?
So the correlation with this sort of human-derived behavior was happiness prediction was fantastic. But I guess that's a sort of a one-dimensional view of what's in the insula. Are there other dimensions of activity? Does it reflect other parts of the history of the reward, of the choices?
Yeah, the Insula is a fascinating, fascinating place. It's kind of infamous for lighting up during every single task ever in fMRI, which Bud Craig, who wrote a book, How Do You Feel, I think, or something like that, kind of likened to if there was a seat of like conscious awareness, that's exactly what it would do. The Insula has a really interesting organization as well. So it is thought to be anatomically the site at which we're getting interoceptive kind of input from the body, as well as what's happening kind of processed by the brain. So the Insula is doing all sorts of things. I think that is kind of at the heart of questions of mood in particular, right? We know that these different experiences are coming in through different senses, like an emotional video is coming in through your eyes, music is coming through your ears, this is like reward is coming in through the reward system, questions about like, where is all of that integrated, right? And are there different integrators in each of the sensory systems? Anatomically, right, we just don't know much about the insula. Like I said, for the primate, this is like one of the only data sets that exists. It's crazy. So if anything, I hope this motivates like more experiments.
But even in the data you do have, right, there are, you've taken this particular exponential weighting and made a prediction from that. But you could look at all, and then you showed us a correlation plot of this signal. But you could look for other dimensions of activity in the insular population activity that might correlate with
during
other
the gambling task.
during the gambling
That
task.
kind of goes back to the answer to this. Yes, so we're very interested in how is what's happening during the gambling trials, how is that transformed into mood? So coming up with a coherent explanation that's more coherent than just giving you a table of bean counts, and that sort of thing. This many neurons were significant. That's not very satisfying.
Let's break for coffee and back for the last session at 3:35.